welcome to this additional and special seminar. So this is the first of the seminars under TIACA. So as you would have seen from the university announcements, is that TIACA, the Centre for Cancer Research, has been acknowledged as one of the 6 university research centres. And so it's an honour that our first seminar is delivered by Kath Bollard. So Kath is one of our trainees in haematology through the Auckland and Starship program. I think it was 10 to 15 years ago, but yesterday you mentioned it was about 20 years ago. I arranged for Kath to do a fellowship with the colleagues in Baylor and Houston around cell therapy, which is emerging technology, with the expectation that she'd come back. She's really become a leader in that area of cell-based therapy, starting initially around the viral targeted cytotoxic T lymphocytes. Kath is now director of around the Centre for Cancer and Immunology Research at Children's National Hospital in Washington, and is really a sort of a leader in this area. And so I was going to talk today about sort of cell therapies and where that's at. So thank you, Kath, and thanks for taking time on your leave to come talk to us.
You're welcome. Thank you for inviting me. And yeah, it's been 22 years, Peter. I know you haven't aged 1 second, but the rest of us have, I have to tell you. So I'm going to talk I did actually speak at the hospital yesterday about the state of cell therapy. So today is actually really talking about our cell therapy program and the advances within the field. I do have disclosures both in the CAR T cell space and in the antigen-specific T cell space. So as I was saying yesterday, you can choose to believe what I say or not, but I do need to disclose these potential conflicts. For those of you who are not all, T cell therapy lovers like myself, I just wanted to summarize what we're going to be talking about today. So we're going to be talking a bit about chimeric antigen receptor T cells. So these are the so-called CAR T cells, which have got a lot of traction in the especially the B-cell malignancy space. So here you have a single chain variable region of a monoclonal antibody, followed by a spacer or a hinge domain, and a co-stimulatory moiety, followed by the TCR zeta chain. So this whole construct here is the chimeric antigen receptor that you then use to gene engineer a T cell, or as you'll hear, other types of immune cells to recognize the tumor cell like the antibody would. So the recognition domain of the monoclonal antibody, the single chain. So it recognizes it like an antibody would, but kills it like a T cell. So the advantage of this strategy is you don't need the major histocompatibility complex. The disadvantage of this strategy is your antigen has to be extracellular so that your chimeric antigen receptor antibody recognition domain can recognize your target. And this becomes problematic in the solid tumor setting, where there's only a limited number of extracellular targets for these tumors. The other flavor, if you like, that we're going to talk about today is the multi-antigen specific T cells. So here, this is technically an existing population of T cells in healthy donors and in patients that are recognizing tumor-associated antigens. These antigens can be intracellular, so they're chopped up and then they're presented on the surface. Little pieces of them are presented on the surface by the major histocompatibility complex that is then recognized by the T cell receptor. So the advantage of this approach is that you can generate a product that, unlike the CAR T cell approach, can recognize multiple epitopes or antigens in a single product. But the disadvantage is it is MHC restricted, and many, especially solid tumors, downregulate the major histocompatibility complex on their surface to evade the immune response. So this is just sort of summarizing at a very sort of low tech level how the T cell would typically recognize a tumor or a virus infected cell through the T cell receptor via the MHC complex on the tumor cell or the target cell that has this little epitope or piece of antigen presented in the context of the HLA molecule. Once you get that engagement, you also have to engagement of co-stimulatory moieties, which then allow the T cell to become activated and release perforin granzyme B to kill, ideally, the tumor cell. So as I said, this is MHC restricted. So another cell type that we'll be talking about today is the natural killer cells or the NK cells. And the advantage of these cells is they do not need MHC on there to recognize their target. In fact, that's how NK cells don't kill healthy cells, because healthy cells do express MHC class 1 molecules. So in the setting of a tumor cell not expressing MHC, this is the perfect setup, if you like, for the activating receptors on NK cells to recognize activating ligands on the tumor cells without needing the major histocompatibility complex. And this allows this recognition and killing by NK cells. So there's multiple different strategies that we can use to harness the immune system to kill tumor cells. But, it's really stacked against us in many ways. Tumor cells use multiple strategies to overcome the immune system. And, I just talked about downregulation of MHC. They also downregulate antigens that different T cells might be recognizing, and they secrete multiple factors that have immunosuppressive effects on the T cell or the NK cell response. And furthermore, these cytokines that get secreted also attract other immunosuppressive cell types like regulatory T cells and tumor-associated macrophages that can all create this highly immunosuppressive microenvironment. So this is a Hodgkin-Reed-Sternberg cell, but most human cancers have these, employ these multiple evasion strategies. However, you can sort of argue, well, if a tumor cell needs this many tumor immune evasion strategies to overcome immune attack, by just knocking out, one or two of these, it might stack more in favor of the immune system to allow these cells to attack the tumor. And so I circled, potential strategies that can be, targeted by antibodies and gene engineering strategies. And we will be talking about TGF-beta during the course of this talk, because it's secreted by most human cancers and has devastating effects on T cell and NK cell cytolytic activity in the tumor microenvironment. So starting with antigen loss, and this is a commonly employed strategy by tumor cells, and this is just an example of the EBV-associated cancers. So this is the highly immunogenic type of lymphoma that develops in patients after transplant. These tumors develop in patients with no effective T cell immune response, so they actually don't have, it's one of the few tumors that doesn't really employ tumor immune evasion strategies, because it doesn't really need it, because it developed in the presence of an absent immune system. But when you start moving to these types of EBV-associated cancers that develop in the immunocompetent host, you can already see downregulation of these immunogenic EBNA antigens is a potent strategy. So this was work that I first started when I went that went to Baylor over 2 decades ago to try and target these less immunogenic antigens expressed by these so-called type 2 latency tumors, in particular non-Hodgkin's lymphoma and Hodgkin lymphoma. And it was really built on the success of using T-cell therapies for this sort of immunogenic tumor. So moving with this type 2 latency targeting, we had to target the LMP, or the latent membrane protein antigens. And back in the day, we used gene engineering strategies of our antigen presenting cells, which included monocyte-derived dendritic cells and EBV transformed B cells, or lymphoblastoid cell lines, to stimulate and expand a T cell population that would be enriched for specificity to the LMP antigens expressed by the adenoviral vector here. So depending on the products that were manufactured, we either had products that were specific for just LMP2 or for both LMP1 and LMP2. And we use these T cells in a phase 1 setting to treat patients with relapsed refractory Hodgkin lymphoma and non-Hodgkin lymphoma of all age groups, as you can see here. This was an autologous product, so made from the patients themselves. We did not give any additional chemotherapy to these patients, so they literally just received these 2 doses of the T cells 2 weeks apart. And with that, you can see these in patients who had relapsed refractory disease, so in the large part chemo refractory disease, we saw 12 out of 21 complete remissions, which led to a 50% disease-free survival at 2 years in a patient population based on their risk factors and how heavily they'd been pre-treated, would be more estimated to be, in the order of about 30%. But even more promising was the patients who we treated as adjuvant therapy, where we saw a 90% disease-free survival at 2 years. We looked for expansion of the T cells in the peripheral blood as a sort of a biomarker strategy, and certainly in the responders, it looked like there was more expansion of these cells in the peripheral blood of the responding patients, but it didn't meet statistical significance. So we then looked to see if we could see evidence of antigen spreading. And this was something that hadn't really been done in the adoptive T cell therapy space before. People looked at this in the cancer vaccine space, but here, we were giving EBV targeted T cells to patients, and what we see here is in the responding patients, we're seeing expansion of T cells recognizing non-EBV tumor-associated antigens, predominantly the cancer testis antigens that were present on the patient's tumor, but not obviously recognized on purpose by the T cell product. So this concept of antigen spreading is something that we're now looking at in more detail in these patients that get these types of antigen-specific T cell therapies. Because we didn't use prescribed lymphodepletion or any other chemotherapy in combination with these approaches, we saw very little in the way of toxicity and no severe toxicity. We did see accumulation of the T cells at disease sites, and we did see, as I said, anti-tumor effects. And we published this paper in JCO in 2014. We went on 4 years later and showed this product could be used for patients after allo transplant. So these are some of the worst patients, which are patients with EBV positive T cell disease. These patients generally do very poorly post-transplant, usually with a 2-year overall survival at more in the order of 20 to 30%. And you can see here in these patients, they had a 2-year overall survival of over 60%, and even better in the patients with the EBV positive B-cell disease. So again, very little in the way of toxicity, and so really is an attractive way to keep these patients in a durable remission, these highest risk patients in a durable remission after allo transplant. So we sort of then are sort of moving this technology forward. This technology platform was licensed by a called Cellmedica, who are looking at this in the NKT cell lymphoma space. Keeping with the virus-associated cancer theme, we have a lot of activity, including 4 clinical trials that are ongoing, utilizing HIV-specific T cells, especially for patients with HIV who also have underlying hematologic cancers. We have preclinical work targeting HPV, and then obviously we still have work going on in the EBV lymphoma space. And one strategy that I just wanted to show you, because it leads on to what we're doing in the solid tumor space, is genetically engineering these EBV T cells or LMP T cells to overcome TGF-beta. So as I said, TGF-beta does have pretty dramatic effects on T cell function in vivo and in vitro. It decreases T cell proliferation and has quite drastic effects on their cytolytic activity and ability to release cytokines in response to antigen. So yeah, back in also in my Baylor days, Claudia Rossig and I manufactured a retroviral vector expressing a dominant negative receptor. And this dominant negative receptor we actually got from Joanne Massagay at Sloan Kettering, which had a stop codon introduced at the transmembrane domain. So everything else extracellularly looks exactly like the wild type receptor. So the TGF-beta binds the receptor normally, but because of the stop codon, there's no downstream signaling. So in other words, here TGF-beta creates a quadrimeric complex with 2 R1 and 2 R2 receptors. So our dominant negative receptor is on R2, but if you've got one piece of your receptor not working, the whole complex fails to work, if you like. So in the normal setting, you have this binding of the 4 receptors together, you get ligand binding, and that results in phosphorylation of the R1 to R2 receptors, and then phosphorylation of the SMADs, which then translocate into the nucleus for signal transduction and all the effects of later effects of TGF-beta. So none of this cascade happens if you have this one dummy receptor within your complex. So that's why they call it the dominant negative receptor. So we used the dominant negative TGF-beta receptor then to gene engineer our LMP T cells like we made for the original trial. So we basically transduced the cells just 1 week before we froze them to give to the patients. So we saw pretty good over 70% transduction efficiency of our T cell products. And when you add exogenous TGF-beta to the T cell products in the non-transduced setting, you of course do see phospho-SMAD appearing by Western, same with the mock transduce, but because the dummy receptor blocks all downstream signaling, you don't see detection of the phosphorylated SMAD2, but you see SMAD2 unphosphorylated in all the settings. Furthermore, the phenotype of the cells did not change, and I should just clarify that all the products that we give generally have about 20% CD4 T cells, cytolytic CD4s in them, and the rest CD8s, with about 20% T cells that express the so-called central memory phenotype, which we think is important for long-term persistence in vivo. You can see here that the expression of the so-called markers of exhaustion perhaps is slightly higher in the transduced T cells, but functionally this is probably because they're activated rather than being functionally exhausted, because they still are polyclonal, as show polyfunctionality in the presence of antigen. Their specificity is also preserved post-transduction with both LMP1 and LMP2 specificity in the transduced T cells compared to the untransduced. So furthermore, as I said, TGF-beta on T cells has causes decreased proliferation of our T cells in vitro and in vivo, and this is a thymidine uptake assay showing in the presence of exogenous TGF-beta a decreased proliferation, which is preserved in the transduced product when you add TGF-beta here. Similarly, when you add supernatant from tumor cells that we know are rich in TGF-beta, you see the same phenomenon. So this trial using the TGF-beta resistant T cells, I won't belabor the years and years of giving these cells to mice first to show safety and efficacy in the murine models. So this is at the clinical trial stage with treating 8 patients, all with Hodgkin lymphoma, all had received previous chemotherapy and did not want additional chemotherapy, so they just received the T cells alone. One patient was relapsed post-allo, and in this setting, the T cell product was made from his brother. So this shows that because we can now really detect the gene-engineered T cells in the patient, we were seeing long-term persistence of the gene-engineered T cells in the peripheral blood of this patient, which correlated with complete remission. His disease was predominantly around in the sternum area, and he has now had a durable complete remission for over 7 years with no additional therapy. Similarly, with this woman, she's didn't have a complete response with the LMP T cells alone, and when we gave her the TGF-beta resistant T cells, she had a complete remission and has always also been in a durable complete remission for over 7 years. So overall on this study, we did have those 2 CRs, and we had 1 continued complete remission in a high-risk patient. We had 1 partial response and 4 stable disease. And so, this looked really encouraging as a therapy, but of course, with the advent of the checkpoint inhibitors in the Hodgkin lymphoma space, it wasn't something, we thought would get much traction in the Hodgkin lymphoma, field. So because of that, we sort of moved to focus more on the solid tumor setting, where we know TGF-beta has a real issue in terms of having devastating inhibitory effects on our immune cells in the tumor microenvironment. So I'm going to talk about how we're sort of expanding to the solid tumor space, including pediatric solid tumors and brain tumors, and how we're utilizing NK cells or natural killer cells as well in this setting. So the natural killer cells, this was a project that we actually started probably about 15 years ago with E.J. Spall at MD Anderson. because she has this large cord blood bank, and we wanted to use cord blood as a donor, as sort of an off-the-shelf donor source for NK cells, because NK cells as well are an attractive off-the-shelf strategy, because unlike T cells, they have a much less propensity to develop, elicit a graft versus host response. So we developed a strategy for expanding these NK cells from these relatively small cord blood units that would be useful for an off-the-shelf use. Plus, we wanted to gene engineer these cells to render them resistant to TGF-beta, because we know that is such an important immune evasion strategy. And we wanted to target neuroblastoma because, A, it's a tumor that is amenable to NK cell attack, but uses TGF-beta secretion as a way to fend off the NK cells from its attack. So this was work that Rachel Berger, who was a grad student in my lab, took the challenge on. And also Russell Cruz and his grad student, Alison Powell, took the challenge. Rachel took the challenge for neuroblastoma, and Alison looked at this in the medulloblastoma setting, because again, medulloblastoma is also a cancer that uses TGF-beta secretion as a potent immune evasion strategy and is amenable to NK cell attack. And so you can see here in the medulloblastoma setting, there's just using NK cells against the medulloblastoma cell line with TGF-beta, you see decreased killing of the tumor cells, but in the transduced cells, there's no such decrease. And actually, what we see is the transduced NK cells can act as a sink for TGF-beta, which could be a real attraction using these cells in vivo, where you now see the sinking effect of the TGF-beta from the tumor microenvironment that may allow other immune cells to do their job. So Alison in that previous study just used our standard dominant negative receptor that we'd used in the lymphoma patients. But Rachel sort of wanted to see if we could turn the negative signal of TGF-beta into a positive signal. So we created a new construct in addition to our what we'll call the legacy DNR, where we added a DAP12 signaling domain. So now when you get TGF-beta binding of the dominant negative receptor, instead of the receptor doing nothing, you now have activation because you've got activation of DAP12, which leads to increased DAP70, which ultimately leads to NK cell activation downstream. So turning that sort of negative inhibitory signal of a TGF-beta into a sort of a positive activating signal for the NK cell. So we compared the two types of constructs side by side. So we saw, very good transduction efficiency of our NK cell products in vitro. Sure enough, the cells that were transduced with the activating receptor did have increases, in particular of phospho-AKT, indicating you were seeing the effects, downstream effects of the DAP12 signaling. So she then put them in mice in a neuroblastoma model. And you can see here that the conventional DNR transduced NK cells are actually do very, very well in vivo compared to your mock transduced NK cells or your untransduced NK cells. But it did seem like the NK cells transduced with the activating receptor did seem to have the best effect in this in vivo model. And when she did ddPCR assays to look for the persistence of these NK cells in the mice, it did appear that the NK cells transduced with the activating receptors had a higher persistence than the NK cells that were just transduced with the dominant negative TGF-beta receptor. So that probably contributed to the improved outcomes in the mice. So that study at the moment, we're sort of looking at translating this to the clinic, and it's really sort of about, well, what's the best model or the best tumor to start this from? And so, you know, that was the work with with neuroblastoma, and we also simultaneously started to move to adult brain tumors, so beyond medulloblastoma. And in part, this was due to when Biden was still a vice president and his son died of GBM, they put out the NCI Moonshot program, and we were successful at competing with this, and we had put a consortium together with a group at Hopkins and at UNC to develop cell therapy strategies for GBM, which, as many of you know, is a really devastating brain tumor that has universally poor survival and poor responses to standard of care radiation chemotherapy. So we wanted to target, sorry, I didn't realize this was animated, target B7H3, and in part this was because, you know, this gene is really expressed although in normal tissues, in most solid tumors, and the protein is preferentially expressed only in tumor tissues. And so really is a great opportunity as a target and as a target for chimeric antigen receptor technology, because it's extracellular and there were single chain antibody fragments available that could be used to target this antigen. The gene itself does sort of downstream, it's part of the immunoglobulin superfamily and participates in the regulation of T-cell mediated immune responses, and generally has immune inhibitory functions. And its expression on tumor cells generally correlates with a poor prognosis. And importantly, for our purposes, is overexpressed in GBM. So we wanted to, you know, look at it as a potential target for the CAR T-cell therapy. So as I said, what we needed to do is have a CAR where the single chain variable region of the monoclonal antibody would recognize the B7H3, and then you have your hinge and co-stimulatory moiety and your TCR zeta chain. So then this CAR was cloned into a retroviral vector system. So now your T cell expresses the chimeric antigen receptor in addition to the a native alpha beta TCR, and so this T cell is now able to kill the tumor cell like an antibody, but recognize the tumor cell like an antibody, but kill it like a T cell. So this CAR was generated by the group at UNC, so Barbara Savoldo and Gian Pietro Dotti, and they evaluated it initially in a using B7H3 CAR T cells in a GBM preclinical model. And you can see they evaluated two types of CARs, one with a CD28 co-stimulatory moiety and one with the 4-1BB co-stimulatory moiety, and both performed equally as well in this in vivo setting. So we wanted to, as part of our Moonshot grant, build on that work and use NK cells instead, just mainly for all the advantages of NK cells and in other, in other realms. One of the big drivers of this was that we could potentially use this product in an off-the-shelf setting. And if we can make cell therapy more like a drug that you can get off the shelf, that'll really help broaden the applicability of what we're trying to do. So this was work done by Kajal Chaudhry, who actually had done done her PhD in Australia and came to us for a postdoc. And she used both peripheral blood and cord blood derived NK cells and gene engineered them to express the B7H3 CAR, and within a 12-day manufacturing protocol. So stimulating the NK cells early, then transducing them, and then expanding them to numbers that would be applicable clinically. So she showed here that these cells after transduction in general do expand, and you can get high transduction efficiency of over 90% in these products. She did show, as I said, that TGF-beta does have, this is different. She showed that, sure enough, the B7H3 CAR NK cells do kill the tumor cells. So these are adult GBM cell lines better than NK cells themselves. So understanding that NK cells do have some activity on their own against some of these GBM cell lines. So you can see the potency of the CAR over the untransduced NK cells. We also have access, we have a large pediatric brain tumor program at our place, and so we also showed that there's good killing of primary pediatric GBM cell lines with the CAR transduced NK cells. Furthermore, you can see good cytokine, in this case interferon gamma secretion in the presence of tumor when you use the B7H3 CAR T cells compared to the untransduced NK cells. But as I said, GBM is one of the many human cancers that also secretes TGF-beta. And unlike in T cells, TGF-beta's effect on NK cells, it doesn't affect their proliferation, but it does have profound effects on their cytolytic activity. So you see marked decrease in the ability to recognize their targets in the presence of TGF-beta. So, but there was this sort of school of thought that was going around that, well, the CAR has got all these co-stimulatory moieties and it's, going to make the NK cell more potent. So surely it won't be susceptible to TGF-beta as anti-cytolytic effects. But in fact, that is not correct. So CAR NK cells are just as susceptible to the anti-cytolytic effects of TGF-beta, just the same as non-transduced NK cells. And the mechanism for the reduction in the cytolytic activity of the NK cells is that the TGF-beta causes downregulation of the activating NK cell receptors on the surface. And so here you can see this downregulation of NKG2D expression on both the non-transduced NK cells and the B7H3 CAR NK cells in the presence of TGF-beta, shown in red. Furthermore, there's with DNAM, we see the same effects. So DNAM's another activating receptor expressed by NK cells. So we used the dominant negative TGF-beta receptor that we had used for the lymphoma patients. So it's what we call the legacy receptor. So has that stop codon introduced just at the end of the transmembrane domain. And so we use this construct in a retroviral vector now to do double transduction of our NK cell products, so with the B7H3 CAR and the dominant negative receptor. So now you have an NK cell product that is expressing a CAR that can recognize and kill a tumor cell, but also protecting itself with the dominant negative TGF-beta receptor. So here you can see nice transduction efficiency of these NK cells that are now expressing both the CAR and the dominant negative and the dominant negative TGF-beta receptor shown down the bottom. So with that, looking at the SMAD signaling, so not surprisingly, untransduced and B7H3 CAR NK cells. When you add TGF-beta, you do see phosphorylation of the SMAD. But with your dominant negative receptor NK cells or your double transduced NK cells, there's no downstream signaling. So this shows that the DNR, even in the co-transduced setting, is protecting the NK cell from the negative effects of TGF-beta. So you can see here, this is just showing across multiple experiments that this blunting of the phosphorylation of the SMAD2 and SMAD3 when you transduce your NK cell products with the dominant negative receptor. So what about functionally? So I already showed you this. Without the DNR, you, in the presence of TGF-beta, you see decreased NKG2D, but you don't see that in the products that now have the DNR on it, including the product that has the CAR and the DNR. This holds true for DNAM and CD16 as well. Again, you see complete protection against the downstream effects of TGF-beta when you have the DNR there. So what about the cytolytic activity? I already showed you when the DNR is not there, you get decreased cytolytic activity with TGF-beta, but now you have complete protection with the DNR. And what's interesting across multiple experiments, this co-transduction with the DNR and the NK and the CAR seems to result in increased cytolytic activity of the CAR NKs. So we're now moving ahead. The in vivo studies are ongoing because if that effect translates to in vivo models, that's a further driver of why you might want to use that double transduced NK cell product in the clinic as an off-the-shelf strategy for certainly for brain tumors, but other human cancers like neuroblastoma that is amenable to NK cell attack and also recognizes B7H3. So that's sort of where we're moving with the NK cell space and sort of looking at opportunities probably in collaboration with pharma to move that NK cell product to the clinic. But at the same time, can we now capitalize on this B7H3 CAR and the TGF-beta dominant negative receptor in the T cell setting for solid tumors? And, we are interested in, obviously, in the solid tumor space with CAR transduced T cells, but if I tell you that there have been in the States, I've got a slide you can't read it because it's on purpose, but it's like, and I'm not exaggerating, about 50 trials ongoing in the States trying to use CAR T cells for solid tumors, and the overall responses in these studies ranges from 0% to at best 5% responses. So a lot of energy and money has been put into CAR T cells for solid tumors to little avail. But where there has been some success in solid tumors is the so-called TILs, so the tumor infiltrating lymphocytes that generally recognize usually cancer testis antigens expressed by solid tumors, in particular melanoma. And so because of that success, we got very interested in expanding T cells specific for these types of cancer testis antigens or other tumor-associated antigens. And we started in the blood cancer setting, and I talked about this yesterday, so I'm just going to summarize what we've done in this setting and then show you what we're doing in solid tumors. But, the reason we picked the antigens that we targeted was because we started in the blood cancer setting. And there's many, potential targets that overlap with multiple different cancers. And, some of the favorites are WT1, the MAGE antigens, NY-ESO, SSX, and survivin, and then PRAME is really stacking up to be a very ubiquitously expressed antigen for targeting by the antigen-specific T cells. So these publications have all sort of really come out in the last 2 years, and they're all actually from either my group or from Helen Heslop's group at Baylor College of Medicine, but these are all in the blood cancer setting, and you can see it includes myeloma, Hodgkin lymphoma, non-Hodgkin's lymphoma, AML, and ALL. And the idea here is we're using overlapping peptide pools for the tumor antigens you want to target. So you basically have the peptides spanning entire antigens. So the peptides are generally 15 amino acids long, overlapping by 11 amino acids, and it spans the entire antigen. So irrespective of your HLA type, we should be able to expand a product for you. And in general, we're using between 3 and 5 antigens to pulse our dendritic cells. We generally stimulate the T cells twice in the presence of cytokines to expand a multi-TAA specific T cell product. So this was the product we took to the clinic for the solid tumors. Because we sort of had started in the blood cancer setting, it was easier to work with the FDA to just use the same antigens that we'd used in that setting. But actually, most of these antigens are expressed by the pediatric solid tumors we wanted to target. So it was focused on WT1, PRAME, and survivin. So these T cells, they're autologous. These were the patients we treated. They were very poor prognosis, predominantly pediatric solid tumor patients with osteosarcoma, neuroblastoma, Wilms tumor, Ewing's sarcoma. This was an adult patient with soft tissue sarcoma referred to us from Hopkins, and then we had rhabdomyosarcoma. So multiple rounds of therapy before they got to us, as you can see here. And this was the swimmer plot. We did not use, again, we didn't use prescribed lymphodepletion, so this is just with the T cells alone. And by RECIST criteria, we didn't see any objective CR and PRs, but as you can see here, there are some prolonged so-called disease stabilizations. And because most of these were pediatric patients and the tumors were in hard to get places, they did not have follow-up biopsies. So many of these patients may be in a more remission state than we think, because what we're following is radiologic only, and we don't know the viability of what we're looking at necessarily. So this certainly was promising, and certainly if you looked at the dose level 3 group, they seem to have better progression-free survival compared to their previous therapy. So it was certainly an encouraging study. Furthermore, we were able to show that the tumor antigens were indeed expressed by the majority of the tumors that we were targeting. The numbers were too small to sort of see if this expression correlated with response. But I think from our point of view, it's encouraging that most of these antigens were expressed, and we know we can use demethylating agents in these patients to upregulate certainly the expression of the cancer testis antigens in these patients that also might augment the anti-tumor effect. Similar to what we saw with the lymphoma patients way back, we not only saw this concept of antigen spreading in our responding patients across multiple patients who had durable, stable disease status, but we used TCR sequencing to show that so here's a classic example of a patient who had a dramatic antigen spreading response at 10 weeks post T cells, and that correlated with an expansion of T cells that we detected by alpha beta V beta TCR sequencing. So we're looking here at the T cells that were present in the product, but not present pre-infusion. So these are, if you like, unique T cell clones that we were tracking over time in this patient, but across all the patients. And the fact that this correlated nicely with the antigen spreading response seems to suggest that the two might go hand in hand. So, you know, obviously this is not a home run therapy at all and, you know, I think the autologous products can be problematic, especially when you're trying to manufacture them from patients who have heavily pre-treated. So, you know, one of the opportunities we have here is by gene engineering those antigen-specific T cells. And so to that end, Martin Poulet and I successfully competed for a Cancer Grand Challenge Award that was a $25 million award that one of the Cancer Grand Challenges last year that came out was targeting pediatric solid tumors. So our Cancer Grand Challenge program is entitled Next Generation T Cells for Childhood Cancer, and has 18 investigators across multiple countries. I didn't make that slide, by the way. That's this Grand Challenge program has 6 work packages, but our work package 5 is really building now on the B7H3 CAR, and this time we're collaborating with Robbie Meisner's group at Stanford, who had made a different B7H3 CAR, and then our dominant negative receptor technology. And then building on the TAA T cells that we had given to the pediatric solid tumors that I just showed you. And we've decided to keep things simple by just using PRAME-specific T cells. So now what we want to do in 3 clinical trials, one run out of Children's and in collaboration with CHOP at UPenn, and then at UCL in London, and then at Stanford, we'll use the same gene engineered platform with the B7H3 CAR and the dominant negative receptor, but in the study we'll transduce the PRAME specific T cells. So these T cells will recognize the tumor through the CAR with the B7H3 and also through their native alpha beta TCR with the PRAME targeting. In tandem at UCL, they will just use non-specific T cells that are expressing the CAR and the dominant negative receptor. Same tumor types though, rhabdomyosarcoma and other pediatric sarcomas, so it'll be run in tandem to see, you know, if there are any differences there. And then the trial at Stanford will use the same product used at the UCL trial, but they'll inject it intracranially for patients with the poorest prognosis pediatric brain tumors, including DIPG and other refractory medulloblastomas. So I usually end my talk with this sort of new dawn for cell therapy, and we're staying right on the waterfront today, and I was hoping to take a picture of the sunset for this, but I don't know if you guys were up early enough to look at the sunrise, but it was completely foggy, so I had to download something from the internet instead. So, you know, where are we sort of going with cell therapies, you know, beyond not just the liquid cancers, you know, understanding that it's still not a home run with CAR-T for liquid cancers. You know, there's definitely opportunities still with multiple myeloma and definitely with the myeloid leukemias where the CAR-T cells have shown very little activity. There may be more activity with the tumor antigen specific T cells, and that's sort of work that we and the Baylor group are ongoing. Certainly there's opportunities in the solid tumor setting with NK cells and TILs and potentially CAR-T if we can get the right targets and the right ability to overcome the tumor immune evasion strategies. And then there's also opportunities in the non-malignant space. So what we're doing for the blood cancers is, really trying to broaden the antigen-specific targeting. So using antigen-specific T cells as a platform for gene engineering with the CARs. And so this is just sort of an example of how we're trying to broaden to other cancer testis antigens, including the MAGE and the SSX antigens. I think what we really want to do in that setting is use these sorts of therapies to enhance our outcomes after allo transplant in particular, to really get a safer, durable anti-tumor effect in these patients who require an allo transplant. To that end, we've also, this is in collaboration with Eric Yvonne, who's at GW now, trying to actually make a CAR that is more like able to recognize the cancer testis antigens. So this is an example of a CAR we've recently made where actually the CAR recognizes SSX, which is an intracellular antigen, but it recognizes the MHC peptide complex. So the antibody region recognizes this whole complex. So it is in an MHC restricted setting, but it performs like, it's constructed like a chimeric antigen receptor. So this is sort of a novel type of CAR to move forward with. And this CAR in vitro, the in vivo studies are ongoing, is really potent at clearing leukemia cells in vitro, unlike the when you use leukemia cells that are not HLA-A2 positive. So, really potent there, and we will sort of see what we can do as we move to the clinic. And what we'd like to do is transduce the T cells with that CAR, and then also with the dominant negative TGF-beta receptor. So that's one space we're working on. As I said, we're working on the B7H3 CAR DNR approach for the cancer grand challenges. We have an active brain tumor program using tumor antigen-specific T cells. One of the more recent protocols that we're just in the process of getting FDA approval for is this is Brian Rood and my labs just published last year, this ability to, with Brian's group, doing proteomics on the tumor cells, and he's focused on brain tumors, identifying novel tumor-specific antigens, and then we generate the T cells specific for those tumor-specific antigens. So it's really a truly boutique therapy, so designed for that individual patient. And we're having a few regulatory discussions with the FDA at the moment to get that to the clinic. As on the opposite side of the spectrum, that's very boutique therapy. And on the opposite side, we've been able to successfully give off-the-shelf virus-specific T cells to patients. So we now want to see if we can give off-the-shelf tumor antigen-specific T cells to patients. So this protocol was approved by the FDA, and we just treated our first patient last week with this off-the-shelf therapy. So that will also be quite interesting to see what happens with that. In the non-malignant space, there's a lot going on in the virus-specific T-cell world. I've just highlighted the areas that we're focused on currently. There's a lot of activity in the cell therapy space with regenerative medicine, with iPS cells and mesenchymal stromal cells. Our program currently has a MSC protocol where we're giving cells through the cardiac bypass machine to pediatric patients after cardiac surgery as a neuroprotective agent. There's also, we don't do this, but there's a lot of work going on in Treg space for organ tolerance, and Adora Lynn and my group is looking at B-regs in asthma and food allergy as a potential therapeutic strategy. So I think, you know, the future is really looking bright in the cell therapy space. I think obviously most of the activity is still going to be in the cancer space, where we do have this opportunity to also combine with other immune-based therapies, including checkpoint inhibitors and other antibody strategies. So I think, you know, what we're struggling with now as a field is, you know, yes, there's some pharma interest, certainly in the CD19 CAR T cell space, but how do we get beyond these boutique centers? And so our group has moved several of our products to multi-center studies through different cooperative groups and consortiums, as highlighted here. But it, you know, and honestly, it's a painful process. So it's really about trying to change people's expectations of immunotherapy and what it can do as a therapeutic modality. And we have to be able to convince pharma that this approach is economic and is worth their investment beyond, you know, the sort of traditional drug model. And, you know, I think where it's different from the from conventional drug development model is, you know, we have to be involved in this process longer, because you know, it's, if you think about it, you know, it's from the patient or the donor to the lab to the regulatory environment to the clinic, and then, you know, going back to the lab, you know, learning what your cells are doing in the patient, and to really be able to develop your next super and better product. And so I think, you know, this is where our bar is for this sort of cell therapy space. We have to be able to show a signal in phase 1, otherwise, you know, there will be no interest to take it further. Your platform has to be broad, so that it's not just targeting a very boutique set of diseases. It has to be made simpler and more easily sort of transportable. And, you know, we as a field in the cell and gene therapy field are still highly scrutinized, and toxicities are generally unacceptable and generally are very newsworthy in the States, and not in a good way. And so it's really quite a high bar to sort of move this forward. But, you know, I think I always do end on this slide, because I think this is sort of where we do sort of want to get to, especially in the cancer setting, to really be able to use these complex biologics at a time when they're going to be safer, i.e. at a time of minimal residual disease, that to then allow these cells to attack the chemo-resistant population, the so-called cancer stem cells, to ultimately effect a cure. And on that note, I want to thank, it does take a village, you can't see the top, and I will just really a big shout out to all my faculty listed down below. So thanks very much, and I know I've gone over, but I didn't know when I started, so thank you.
Thank you, Kath, for another outstanding sort of tour de force.
I did put some more slides in, different slides, right? Some different slides.
Kath's actually here on leave and catching up with sort of family. So I'm so grateful for doing the talk for the clinicians, and then the second for the Centre for Cancer Research and for the faculty. So we've got time for questions. We've got even more time here. Andrew.
Mm-hmm. Mm-hmm. Yeah, and you know that original dominant negative receptor.
Sorry, can you repeat the question so that those people on Zoom can
Maybe not all of
not all of the questions?
No, so we're talking about the yin and yang of TGF-beta and how some tumours actually create their own, if you like, dominant negative receptors. And that original dominant negative receptor was cloned from a human epithelial tumour, actually. So yes, so it is interesting that, yes, so in some cancers, that's right. But it's more, in those cancers, we've also, in some of them, we've shown that TGF-beta is a growth factor for them. And you know, that TGF-beta is still in the microenvironment, which is still having devastating effects on your T cells. So, but yes, I'm glad you brought that up, because that was an interesting way how a tumor cell helped us to develop for the T cell. Yeah, exactly.
Rod Dunbar's got a question online. He says, great talk, Kat. Do you think that multi-antigen specific T cells can induce CR by themselves, or do you need to induce antigen spreading to neoantigens from driver genes?
So the question is, do I think that TAA T cells will elicit tumor effects without antigen spreading? And I think we just don't know yet, Rod. Interestingly, we just completed a study. We need to publish it, but it was sort of negative, but we still need to publish it. So we actually used the TAA T cells in pediatric solid tumors with prescribed lymphodepletion, because, you know, obviously everyone thinks that we need that to make the CAR T cells work. And it did blunt the antigen spreading effect, and we did not see any objective anti-tumor responses. So it was no better than the study we did without prescribed lymphodepletion. And so, you know, I am worried that, yeah, you're right, that probably you do need the antigen spreading effect, and definitely that was another reason that drove us to look at gene engineering opportunities for the TAA T cells to enhance their function, because obviously just using prescribed lymphodepletion is not going to be the answer there.
Andy Wood, who's online, says, thank you for the exciting talk. Even using the double transduced NK cell, what ratio of effector to target cell do you think is achievable in solid tumours, particularly when chemo and surgery leave a residual mass rather than MRD setting? If you click on Q&A.
Okay, yeah, yeah. You know, I think that's really hard to know about what the ETA ratio is required when we get into the patients. And you know, I think that's why, you know, we're looking at in vivo delivery, not just intravenous delivery. And, you know, I think for the brain tumors, you heard that, you know, loco-regional administration seems to be the way to go. That's what's coming out of the Stanford group. And we're partnering with different engineering groups to look at, you know, utilizing nanotechnology to sort of get our T cells to where they need to be and keep them there, and so that you do get the optimal ET ratio. But, you know, I take your point. We don't know. Andy, thanks for the question.
Yeah.
Oh, great.
Oh.
Oh, that would have given me another hour. That would be
But I'm just interested in the safety side. We've tried other things, and to get through our regulatory steps, we have to be very small escalating doses by the time we've done it.
correct. Correct.
So how would you do that? If you just go hard from day one, or do you do
Yeah, I mean, we're sort of pushing back more and more with the FDA these days because, you know, we've been there, done that, given sort of homeopathic doses and 9-10 dose levels, and it's just such a waste of time and energy, frankly. So we try and sort of argue our way through it, why, you know, the dose needs to start here and then build in de-escalation if there's toxicities. And, you know, most of the cancers that we're treating, patients have no other therapeutic options. So I've found that for malignant diseases, for adult malignant diseases, the FDA have given us less pushback recently. Where I'm having real problems is pediatric cancers. They, you know, demand that we evaluate in adults first, and, you know, we're saying, well, that's not possible. These are pediatric cancers, and there they make us start with the homeopathy, and you know, these kids have nothing, and you know, some of these diseases like DIPG, you know, most of the patients are all dead by 10 months. So, and you know, 2-year-old kids, I mean, so I don't know, to me, it's about arguing with the regulators. That's my bottom line. Or like, yeah, building in a sort of a de-escalation as well as an escalation step, but try and hold true with your what you want your starting dose to be. I don't know if that's helpful.
Well, it is, but if someone's publishing, like, with a lot of experience publishing, that does help everyone else. People who know so it doesn't help.
Right. And so, yeah, you know, so certainly with the HER2 CAR, that there's some great papers that Steve Gottschalk published from Baylor, where, I mean, honestly, it was yeah, like 9 dose levels, and you can see how poorly the patients did until they got to like dose level 8 or something. You know, I mean, it was craziness. So.
Has you know, because you've looked through and worked through and been a leader in that area, has the regulatory environment changed as it's become more experienced? I mean, I think the problem that Bill's got is that we don't. This is new in New Zealand environment, so there's not actually the authorities that have really got the experience in that area. So has it changed? You know, like you're working with the FDA.
Yeah, no, it's definitely changed. As I said, you know, for the adults, it's changed a lot, definitely. But yeah, there's still work to be done. And, you know, honestly, during the pandemic with the Great Resignation, you know, what I'm now encountering is that, you know, a lot of the more seasoned FDA regulators have left, and now we've got, you know, more junior people, and now they anonymize who your reviewers are. But when you finally figure them all out, most of them were my fellows, because they're, you know, we're in the DC area, so all these, and I'm just like, so, you know, I think it's all about educating. So now the FDA allow us to come and educate them. So I think actually having a close relationship, well, as close as you can get within the rules, with your regulators from an education perspective is so key, because you have to understand you will know far more than them. You know, no one will know more about your product than you know? So yeah, so that's yeah. And, you know, I think that was why I, you know, as Peter knows, I volunteered to serve on the DSMB for the CAR T-cell product that the Milligan are evaluating in people, because, you know, I was like, well, you know, they need to get that through to the clinic. They need that to get that to patients. And, you know, Peter and the group down there were concerned there just wouldn't be the expertise in the regulatory pool to get that through. And I mean, they did, so it's possible.
All right. I think we
yeah.